Hello, everyone! Have you ever wondered how doctors can detect a virus, measure hormone levels, or even diagnose certain diseases from just a small blood sample? One of the most widely used laboratory techniques for this purpose is ELISA, which stands for Enzyme-Linked Immunosorbent Assay. ELISA is a highly sensitive and specific immunological assay used to detect and quantify antigens or antibodies in biological samples. In this video, we'll explore how ELISA works, its different types, and why it has become one of the most important diagnostic tools in modern medicine. ELISA is based on one simple biological principle. Antibodies bind specifically to their target antigens. Instead of using radioactive labels like radioimmunoassay, ELISA uses enzymes attached to antibodies. These enzymes produce a visible color change when a substrate is added, allowing scientists to determine whether the target molecule is present. To perform an ELISA test, several components are required. First, a microtiter plate, which contains many small wells where the reactions take place. Second, either an antigen or an antibody is immobilized on the surface of the wells, depending on the type of ELISA. Next, the patient's sample is added. This sample may contain the antigen or antibody that we want to detect. An enzyme-linked antibody is then added. This antibody specifically binds to the target molecule. Finally, a substrate is introduced. The enzyme converts the substrate into a colored product, producing a measurable signal. Let's go through the basic ELISA procedure step by step. First, the antigen or antibody is coated onto the microplate wells. Next, a blocking solution is added. This step is very important because it covers all the remaining empty surfaces of the well, preventing antibodies from binding non-specifically. After blocking, the patient's sample is added. If the target molecule is present, it binds specifically to the immobilized antibody or antigen. The plate is then washed several times to remove any unbound molecules. Next, an enzyme-linked antibody is added. This antibody binds specifically to the target molecule. Another washing step follows to remove excess antibody. Finally, a colorless substrate is added. The enzyme converts the substrate into a colored product. The color intensity is measured using a microplate reader, which provides quantitative results. There are four major types of ELISA: direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. In direct ELISA, the antigen is attached directly to the plate. A single enzyme-linked primary antibody binds directly to the antigen. This method is fast and simple, but generally less sensitive. Indirect ELISA uses two antibodies. First, the patient's primary antibody binds to the antigen. Then, an enzyme-linked secondary antibody recognizes the primary antibody. Because multiple secondary antibodies can bind to one primary antibody, this method provides greater signal amplification and sensitivity. Indirect ELISA is commonly used to detect antibodies produced during infections. Sandwich ELISA is one of the most commonly used formats. A capture antibody is first attached to the plate. The antigen from the patient sample is then captured by this antibody. Next, a second enzyme-linked detection antibody binds to another site on the antigen, forming an antibody-antigen-antibody sandwich. This method is highly sensitive and highly specific because two antibodies recognize the same antigen. Competitive ELISA works differently. The patient's antigen competes with a labeled antigen for a limited number of antibody binding sites. As the concentration of antigen in the patient's sample increases, fewer labeled antigens can bind. Therefore, the signal produced becomes weaker. This creates an inverse relationship between signal intensity and antigen concentration. This table provides a summary of the four types of ELISA, including their visual schematics, target analytes, and advantages. Photometry serves as the instrumental bridge between biological interactions and digital quantification. The conversion of light attenuation into concentration data is governed by the Beer-Lambert law, which states that absorbance is directly proportional to the concentration of the absorbing medium. The intensity of the color reflects the amount of target molecule present in the sample. The stronger the color produced, the higher the concentration of the target molecule. ELISA has numerous clinical and research applications. It is widely used to detect infectious diseases such as HIV, hepatitis, and COVID-19. It can also measure hormones, including insulin, thyroid hormones, and pregnancy hormone HCG. In research laboratories, ELISA is commonly used to quantify cytokines, growth factors, and antibodies. The food industry also uses ELISA to detect allergens and food contaminants. Because of its accuracy, sensitivity, and relatively low cost, ELISA has become one of the most widely used immunoassays worldwide. All right, let's see how well you were paying attention. Here is your pop quiz. Question 1. What does ELISA stand for? Is it A, Enzyme-Linked Immunosorbent Assay, B, Enzyme-Labeled Immune Screening Assay, or C, Enhanced Laboratory Immunological Sample Analysis? The correct answer is A, Enzyme-Linked Immunosorbent Assay. ELISA, which stands for Enzyme-Linked Immunosorbent Assay, is a highly sensitive and specific immunological assay used to detect and quantify antigens or antibodies in biological samples. Question 2. Which component in ELISA produces the visible color change? A, antigen, B, antibody, or C, enzyme acting on a substrate? The answer is C, enzyme acting on substrate. These enzymes produce a visible color change when a substrate is added, allowing scientists to determine whether the target molecule is present. Final question. Which type of ELISA uses two antibodies to detect an antigen? A, direct ELISA, B, indirect ELISA, or C, sandwich ELISA? The answer is C. Sandwich ELISA is highly sensitive and highly specific because two antibodies recognize the same antigen. How many did you get correct? Let me know your score in the comments below. If you enjoyed this video and found it helpful, don't forget to like, subscribe, and share it with others interested in molecular biology. Thanks for watching. Stay curious, keep exploring, and we'll see you in the next deep dive into the wonders of science.